to produce branches. And if the cytokinin level is low relative to auxin, that's going to promote branching of the roots as opposed to the shoots, so formation of lateral roots. And for example, in tissue culture, like you see here, if you just applied auxin, the cells would just get bigger and bigger, but they wouldn't divide. It's really, you have to have cytokinin in there in addition, and at just the right ratios in order to get normal growth from tissue culture. And these compounds, cytokinins, are called cytokinins because cytokinesis is the name, the technical term for cell division. And as you know, they're well named because that's one of their main effects here. So again, it's really, when we chop off the terminal bud of the plant, we're dramatically increasing the cytokinin to auxin ratio because auxin's dropping dramatically. And that's really what's going on to initiate the development of these lateral shoots, is this high cytokinin to auxin ratio, this increase in the cytokinin to auxin ratio. And so we look at the plant overall. The auxin is, as I mentioned, produced in the terminal bud of a shoot, and it's transported down the xylem through the parenchyma cells. The cytokinin is produced in the root tips at the opposite end of the plant, and it's being transported up the plant by the xylary elements, the tracheids and vessels, which are carrying water and minerals up the plant. So it's being carried through the normal channel in the xylem. And so we get this gradient of auxin and cytokinin within the overall plant body and down into the roots as well that basically sets up the kinds of growth responses that we'd expect in a normal plant. And so it's a really nice example of hormone balance and how you can look at it in a very holistic way here and see how this affects the whole plant's development. So again, cytokinins are involved in cell division and differentiation and in axillary bud growth as opposed to inhibition. All right, so there's another class of hormones that's involved in stimulating growth in plants, and in particular in stems and in leaves, and it involves mostly stem elongation. And these are called gibberellins. And these are compounds that were actually first discovered in rice seedlings that farmers found in Asia that were growing long and spindly and toppling over. It was called the foolish rice syndrome, I believe is what they called it. And Japanese plant physiologists studied this and they realized that these rice seedlings were infected with a fungus called gibberella, the genus gibberella, and that's the basis for the name gibberellins, for the compounds that were isolated there that had a clearly stimulating effect on stem elongation. And so it's really, this is a similar type of effect to auxin, and gibberellins and auxins work in concert in regard to stem growth. Also, like auxins, gibberellins have an effect on, well, I should also mention here that gibberellins are really important in promoting seed germination in certain species of plants. So if there's a light requirement for germination, as there is as a cue for germination in many plants, which helps the plant, which basically is the signal for a plant to, a cue for a plant to germinate when it's close enough to the surface that it can actually successfully emerge from the soil that light's penetrating. These gibberellins will result in initiation of enzymatic action that degrades the starch in the endosperm and gets the ball rolling on the whole germination process. And we use gibberellins a lot in the lab for stimulating germination in plants that we study in the herbarium. And there's a group of plants that I work on a lot that has this light requirement. It's really hard to break the dormancy oftentimes. But if you apply just like 150 parts per million, 200 parts per million gibberellic acid, the things almost burst out of the seeds, and the next day they're really actively germinating. So these really do have a major stimulating effect on initiating seed germination when there's a light requirement. Like auxins too, they promote fruit development in terms of fruit enlargement. And commercially, gibberellins are actually applied to some grapes, Thompson seedless grapes, as a spray. And on the right, you can see the outcome here, where not only are the fruits larger than in where gibberellins weren't applied, but the internodes between these individual fruits are also longer, and that helps to keep the fruits apart from each other, and they aren't tightly clustered, which can promote fungal infection. So gibberellins, like auxins, involved in stem elongation. They're also really important in what's called bolting, which is the response in plants that have sessile rosettes of leaves. A lot of herbs go through their life basically without elongation of the internodes between the leaves. Think of a lettuce, for example, where the leaves are all tightly grouped together at the base of the plant, and then at a certain point in development, they will rapidly produce a flowering shoot. You think of a century plant or an agave or something like that. That bolting response is due to gibberellin. And I already mentioned seed germination and fruit enlargement. There's another class of hormones involved in stimulating growth of the shoot, and these actually look quite a lot like animal hormones, more so than other plant hormones. They're steroids, racinosteroids. So steroids, of course, are like cholesterol. Plants are animal sex hormones. You would have thought that these would have been discovered early on because of their similarity in structure to animal hormones, but in fact, this is the most recently discovered class of plant hormones. The name has to do with the fact that they're discovered in a member of the Brassicaceae, the mustard family, Arabidopsis, which is the model system for a lot of plant biology at this point. And it was found that their function is so similar to auxin that they've kind of gone unnoticed as in terms of their effects. They do promote stem elongation. And that was discovered because some mutants that weren't undergoing normal stem elongation, some dwarfish plants of Arabidopsis, were found to be lacking in racinosteroid production. And if racinosteroids then were applied, they would undergo normal development. So this is a relatively recently discovered group of hormones, but interestingly, they are chemically a lot like some animal hormones. All right, so now there are some hormones that are actually involved in suppression of growth as opposed to promotion of growth. And one of these is abscisic acid. This is a really important plant hormone, often called ABA. And it's very important. It plays the crucial role in seed dormancy. We already talked about dormancy of seeds. So when plant seeds are developing up to the point where maturation of the seed is complete, remember the seed goes into a dormant state, it desiccates, and this is the only stage of plant development in seed plants that's desiccation tolerant. And it's ABA that sets all that into motion. And in order to break dormancy in seeds, we have to remove ABA. And ABA can be broken down a number of different ways in different plants, but if you can remove ABA, you often can initiate germination quickly in otherwise strongly dormant seeds. And we do this in the lab by removing all the membranes away from embryos in the case of members of the sunflower family that are really strongly dormant, and bam, they just start germinating in the light right away. So there are some plants that are deficient in the production of ABA, and some of these are mutants where the seed will unnaturally start to germinate inside the fruits. where ABA would normally suppress their germination. But there are some natural wild plants that have a lack of ABA in their seeds, and this is actually important to their survival, like the mangroves, which occur out in these tidal wetlands, where it's really important to get their root systems established very, very quickly. And as you can see here, here's a seed, and we have the root here that's actually developing prior to the seed even being dropped from the plant. And here you can see a mangrove being established. They have to get established in these tidal mudflats really quickly. And so sometimes it's not a great thing to have ABA, depending on your ecology. All right, so ABA also is the hormone that's responsible for stomatal closure. So once the guard cells have become turgid, of course, the plant's going to lose water from the pore in the stomates. And remember that potassium entering these guard cells allows water to follow by osmosis and results in the turgidity. And what ABA does, so the plant roots are often the first part of the plant that detects water shortage. And the ABA signal can be transported to the leaves from the roots, for example, and this can result in an opening of the potassium channels and membranes. The potassium leaves the guard cells, and the water follows by osmosis. And of course, then as these guard cells become limp, the pore closes. So this is a way that the plants regulate their water balance very finely, and they really do have very fine detection of drought stress, which is a crucial survival mechanism, and abscisic acid is involved in that response. So abscisic acid is involved in the dormancy of seeds, the closing of stomata, and in general, you could characterize its action as being more of an inhibitor of growth rather than a stimulator of growth. And it's the balance between ABA and some of these growth-stimulating hormones that often dictates what kind of growth response is going on in a particular tissue. So ABA is also involved in this hormonal balance we're talking about. Ethylene is another type of plant hormone that is not really a growth, well, it's often involved in plant senescence, that is in responses that will result in growth suppression as well. It is an important signal for leaf senescence and abscission in deciduous plants. Of course, many plants lose their leaves during periods of climatic stress in the winter, in the case of winter deciduous plants, and in summer in places like California where we have a long drought period in the summertime. And the way that ethylene works is to, well, first I want to mention ethylene is a very simple, small compound, and it actually functions in the gaseous phase. And so even though it was proposed as a plant hormone long ago, it took a long time for plant physiologists to really accept that this was a real plant hormone, because it's a very simple molecule and it's also not active in the normal phase. I mean, it's unusual in having this activity as a gas. But anyways, it does act in regard to setting up this so-called abscission layer at the base of leaves and deciduous plants. And there is a small zone of parenchyma cells near the base of the leaves of deciduous plants. Remember, parenchyma cells have very thin primary walls. Well, the ethylene acts to initiate the breaking down of those cell walls and the weakening of the stalk of the leaf, the petiole. And as that's going on, the ethylene also is initiating the formation of cork, which remember has suberized cell walls, making them a nice dermal tissue that's basically impermeable to water, and that will form right to the inside of that, where that abscission layer is breaking down, such that when the leaf finally detaches under its own weight as the cell walls break down or in wind, there'll be a cork layer there that protects that leaf scar, as we call it, and prevents water loss or entry of organisms through that scar. All right, so ethylene is commercially really important in ripening of fruit, and you probably had a lot of experience with the actions of ethylene with fruit. It's often applied to fruit that's been picked way before it's ripe. I mean, a lot of times for shipping purposes, if you pick fruit that's ripe and try to ship it across the country, you're going to have bad consequences. It'll spoil before it gets to market. Pick it early, and when it gets to market, apply ethylene, gaseous ethylene. That's going to initiate the ripening process. And if you want to store fruit that's ripe or prevent ripening from initiating, there are a number of different ways you can prevent the production of ethylene, for example, using CO2 gas or various filters to get rid of ethylene like you see here. These are all really important commercially. So how ethylene operates is it signals ripening, and this ripening process is the enzymatic breakdown of the cell walls. So the fruit goes from being hard to soft because the cell walls break down. And it also converts the starch to sugar, so it results in the sweetening of the fruit. And we also get oftentimes changes in coloration or odor of fruit as a result of these processes that ethylene triggers that helps to stimulate an animal dispersal agent, attractant for animal dispersal. And it's one of the few cases in physiology of a positive feedback. So the production of ethylene initiates ripening, ripening initiates the production of more ethylene, and if you put the fruit inside a closed container, this can get out of control pretty fast. And as anybody knows, once you have one ripe fruit in with a bunch of other fruit, the process can really take off, and one fruit can initiate the ripening of other fruits, for example, inside a paper bag or some kind of closed container. So ethylene promotes various types of senescence. It also promotes programmed cell death. So in the case of, remember, sclerenchyma or certain vascular tissue in the xylem, the tracheids and the vessel elements, those cells are dead at functional maturity. That programmed cell death is triggered by ethylene, and the ethylene is not just involved in killing the cells, but in basically harvesting all the components of those cells and making them soluble and reabsorbed by the plant. So to summarize, plant hormones can have a variety of effects. They can also have contrasting effects. Different plant parts can have different sensitivities to the hormones we talked about. There's differential sensitivity. And hormones often act together, the ratio of them being important in determining the growth outcome, the hormone balance idea. All right, so I just want to conclude here in the last few minutes with plant defense. And this often involves chemistry. So when a microbe like a fungus or a bacterium attacks a plant, it can result in a very local and specific type of response called a hypersensitive response that results in the plant basically walling off that part of the plant with lignin and causing programmed cell death of the cells inside that area that have been damaged, and you're waging all-out war in that area. And at the same time, it's producing chemical signals that move to other parts of the plant and initiate the transcription of defense genes that have a broad array of different defense functions. And so we can get systemic acquired resistance in that way that will ward off a diversity of different pathogens. So if you're being attacked by one pathogen, it behooves you to protect the rest of the plant against that and maybe other pathogens that might be occurring in the environment at that time. And of course, herbivore attack can also be warded off by physical defenses like the waxy cuticle, spines, and other things for in case of larger herbivores. But one of the coolest things about herbivore defense that plants can do is with their chemicals. And there's a lot of these secondary metabolites, these chemicals that are not directly involved in growth, development, or reproduction, the plants produce a huge array of things like alkaloids, terpenes, phenolics. These kinds of compounds are harmful to enemies. They can foul up digestion in the case of something's eaten them. They can kill the animal outright if it's eaten. And in the most amazing examples, that kind of attack can even elicit the production of airborne chemicals, volatile chemicals, that will attract the natural enemies of the herbivore that's attacking the plant, which can be interpreted as a call for help by the plant. Although in the case of the predator of the herbivore that's coming to the rescue, it's not altruistic. It's basically a dinner bell for the predators. So the plant, like a tomato plant being attacked by spider mites, herbivorous mites, will produce a volatile chemical that can be detected then by predatory mites like the one seen here. And they'll come to the plant and eat the herbivorous mites. And various plants have evolved these kinds of responses that have then been basically natural selection has resulted in animals picking up these cues and being rewarded for these kinds of visits. Okay, so that's the end of the course. I hope you've enjoyed it and that you have good luck with the midterm and the final. I'll continue to have office hours on Friday. You can reach me by email to set up an appointment next week if you need to. Please come to the review session. It's going to be in this room at 8 o'clock, 8:10 on Friday, or watch it on webcast if you can't make it. And please consider the other courses that IB has to offer. You've had a great introduction to ecology, evolution, and plant biology. And we have some of the best courses in the nation for this kind of these topics, and I hope you enjoy them. So good luck, everybody. Thank you.